Hello, everyone. My name is Ayan Banerjee, and my presentation is titled, Is Base and Prime Editing the Future of, s- sorry, is Base and Prime Gene Editing the Future of Curative Treatments for Cystic Fibrosis? So, a quick introduction to cystic fibrosis. It's an inherited autosomal recessive disease, and both alleles must be present in the child from the parents for about the mutated gene for it to be expressed, basically. And it's a lifelong disease that leads to lung failure and a really short life, and thus it can be classified into three or five classes, with classes 1, 2, and 3 being the most severe, and that's because of the CFTR gene causing the most severe protein deformation to the classes 1, 2, and 3 patients. So yeah, as you can see on the image on the right side, we have all the chloride ions which are not being able to be regulated in the bottom image right here. In the red, we see that they're not being able to be let out of the lung cells, whereas in the top portion, we see that the proper pathway allows proper CFTR regulate or CF chloride regulation, and because of the bottom, the thick mucus builds up in the lungs, and the patient can stop breathing. And the most common mutation for cystic fibrosis is F508del, which is a deletion of 3 nucleotides, and it occurs in 80% of patients. So different strategies to treat this disease. Right now, the most common we have is the potentiators and these correctors over here. As you can see, they only address the upper half of this diagram, which would be like everything expressed by the gene. Whereas in the next slide, what I'm going to focus on today is CRISPR gene editing, and we see that it attacks the gene directly, therefore stopping all the subsequent steps from happening and restoring normal function. Okay, so quick introduction to CRISPR. CRISPR is a gene editing technique that targets specific sequences and uses a Cas9 protein and guide RNA. The technique introduces DNA double-stranded breaks, and the problem with this is that it can often be inaccurate and carry out unwanted gene edits, which can create harmful off-target edits, which are not good for the patient. So that's why today I'll be investigating these two derivatives of CRISPR-Cas9 called prime and base editing. So first up is prime editing, and in prime editing, the parts used are another inactive Cas9, but this time it's called a nickase, and also that creates a single-stranded DNA nick at the target site. And then we also have a reverse transcriptase enzyme and a PE guide RNA, and they work together to synthesize and repair the gene. And because it only creates one nick and not a full double-stranded break, it's a lot more accurate while still being able to maintain all the editing options. So as you can see here, prime editing can help fix insertions, deletions, and point substitutions. And this is good for F508del, considering F508del is a deletion mutation. And these PE systems can further be optimized by introducing a second nicking guide RNA that can avoid harmful DNA repair mechanisms. So as you see on this slide, the top pathway is just the adding of the new edited sequence by PE. And then as you see this arrow, since we added a new DNA sequence, obviously the top strand will be a bit longer. And in the traditional pathway, as we see in the middle part, the endonuclease, which is an enzyme, will just cut off the excess DNA strand. And then this allows the DNA polymerase, which synthesizes the other side of the other strand, to just synthesize everything downstream. And that can cause these insertions and deletions, which are not good. But in the optimized pathway that the researchers used, we have a second Cas9 nickase over here, and that just allows the DNA polymerase to synthesize one specific portion, which allows for controlled incorporation of the edited sequence. Okay, so now base editing. Base editing also uses an active Cas9, and it's attached to a base pair modifying enzyme, and the guide RNA is bonded to an adenine or cytosine deaminase, and the entire thing is called an adenine or cytosine base editor. And the one disadvantage of this is that these can only convert A to T base pairs, AT base pairs to CG for ABEs, and then CBEs can only do the opposite. So it's only just flipping the base pair, but it's also more precise and accurate than prime editing. And then another advantage is that class 1, which are the most severe, can be most commonly treated with this type of editing, and that will help limit this diagram right here. So on the left side, we see the star, which is the mutation, and as it goes down, the mutation is within the mRNA. And when the mRNA is getting translated, we see the ribosome comes here and tries to make an amino acid to make this CFTR protein, but it's stopped when it sees this mutation. And when the ribosome sees a mutation, it stops translation, and therefore all the mRNA is degraded and no protein is made at all. But when the base repaired mutation is present, we have the mRNA translated and the ribosome continues normally, and we have a fully functional CFTR protein, which again helps to regulate the chloride transport. Okay, so what was addressed? In addition to F508del, N1303K and L227R were also treated by prime editing, which are not treatable by modulators, which are the potentiators and correctors, which make the need for them more urgent. And then also we have the F508del and the G542X, as well as a W1282X and R553X treated by base editing. And basically how they measured the success is they saw if the CFTR protein levels were corresponding to the gene correction, which corresponds to lung function. And like I said, they optimized it by using this special prime editing second nicking guide RNA system, as well as these two ABE8Es and ABE9E base editing systems. And that would turn out to be the most precise and accurate. And they both tested them in different cell types and tested them in different organisms as well, just to get a better understanding. So the data table, we see that all seven rows have substantial mutational correction, functional restoration, and all have really low numbers for the bystander edits and off-target edits. So yeah, both of them had substantial edits above 25%, and base editing statistically was more likely to repair the target sequence and revert mutations. So this is what's used by base editors for the F508del. And since it can't edit F508del directly, it uses other mutations to basically mask it basically uses other mutation to mask the F508del's harm, and even that was edited to 50%. And then accuracy, all of them had sub 1% off-target edits and about 10% bystander edits, which is really successful. And as you can see in this table, functional restoration, the number that's needed for a treatment to be considered successful is 10%, and everything here is much more than 10%. And then, yeah, and then gene editing, like the overall conclusion, I guess, would be prime editing is better suited for the most common F508del, and then other frame shifts as well, and then base editing will work best for the class 1 point mutations. And then just to compare this to modulators, they're a bit better than modulators, and then that could make this more promising for the future. And then also something else that could be considered is a hybrid treatment, which uses both modulators and gene editing, and that could be even more successful, but only gene editing would have to be approved independently first for this to even be an option. Okay, and then just going on to the big conclusion side. Right now, there's 84 million yearly given in the US for cystic fibrosis research, but that can only develop like small prototypes. So in order to keep a steady supply in the clinic, which is what the goal of this paper was, 80% of these biotech costs right now are variable and unpredictable, and that would need to be fixed because investors are not willing to put money in without profit. And because of the capital needed for any new technology, right now, the big conclusion is that gene editing cannot become a primary cystic fibrosis treatment due to current regulations and funding. But in the lab, scientifically, they're both superior. So if conditions were to change, then it could possibly become a cure in the future. And then just some shortcomings of this paper. It did not examine the different methods of in vivo delivery and for base and prime editing, which could disregard the risks and urgency of gene editing, and that could play a big factor into getting it into the clinic. And then also gene damage. Guide RNA can harm the genome and cause scaffolding and strand discrimination in nonsense mediated decay, which was that slide with the two pathways I showed earlier. And then finally, obviously, the last thing about inequitable access, even if it was to make it out of the clinic, out of the lab into the clinic, there could be access only for the very wealthy few. And then also mutational prioritization, like F5 is the most common, so it may just be prioritized, while other people who have other mutations may just be left behind. And then patients, even after all this, if all goes well, patients still may not trust the new treatment because of it's just so new and it's just something so novel. They just may want to stick to the other ones, which could hinder its success. And that's why on the right side I have kind of this yin and yang of like gene editing and all the negatives on the left. I mean, all the positives on the left and all the negatives on the right are the challenges basically. And then just acknowledgements. I would like to acknowledge my project mentor Rami, my showcasing mentor Lolita, and the Polygence program for helping to create this project. Thank you.
Fantastic. Great presentation. If anyone has any questions, feel free to add them to the Q&A section. I think we have a little bit of time, so I will ask one if anyone has any that they'd like to add, feel free. But I was just curious, what was the hardest part of this research process for you?
The hardest part of this research process was analyzing all the different papers because this was my first time ever writing a research paper. So I was like not familiar with the jargon of like all these scientific papers and they were really long. So the hardest part was in the summer, just like decoding everything they were saying and then synthesizing it into my own paper. And like I went from basically not knowing anything about gene editing to pretty much becoming a little expert on it just by reading these papers. But that was a lot of hard work. So
Yeah, for sure. Well, thank you. Thank you for sharing that.